Thank you very much. Thank you for inviting me as a kind of exotic speaker in this session. I have no knowledge, no particular knowledge in ALS. So the presentation I'm going to show, I don't know if I have to press on something or not, okay, is completely focused on drug development from the point of view of the European Medicine Agency. I have to also apologize to the US colleagues because this presentation is strongly oriented to the situation in Europe, which is significantly different from what happens in the US. So I will tell you how this medicines agency look at development, including biomarkers nowadays. So I think it's very clear that these new developments based on biomarkers are really welcomed by the agencies, and they are even promoted. They, in some cases, like in oncology, they do constitute a real paradigm shift in how these agencies are approving drugs on now much smaller groups of patients, but much more selected, much better selected. So the biomarker is not always the same. You have to be very clear if you're talking about predictive or prognostic biomarker measured before starting the treatment, or if we're talking about the pharmacodynamic marker measured during treatment or at the end of treatment, and some of them may be, of course, surrogate endpoints. So the better the patients are selected, the happier the agencies will be. There are two types of scenario to discuss the situation with the European agency. The first scenario is, I would say, globally a companion diagnostic approach. So a company has a new product and wants to develop it in parallel with a biomarker. And this is a complicated story in Europe, unfortunately, I would say, or fortunately, because on one hand, the European agency is only concerned with drugs, not with devices, so that the company has to develop their devices according to different standards, which are not part of the European Medicine Agency, which are part of a different regulation on in vitro diagnostic in general, and in vitro diagnostic devices, in vitro devices in general, and diagnostic devices in particular. So the development has to run in parallel, and the medicine agency will only look at the global picture at the end. So this is very different from what happens in the US. The US guideline, which has just been out for a year now, defines a companion diagnostic as a diagnostic device that provides information that is essential for the safe and effective use of a corresponding therapeutic product, so you cannot do without it, or you have a different development plan. The European Commission is now working on a revision of the Medical Device Directive, which will be implemented across Europe, not next year, because there will be elections at the parliament, so we have to wait for the elections, as in every country, but probably in 2015. So that would be a device specifically intended to select the patients. Again, select the patients with a previously diagnosed condition or predisposition as eligible to a specific therapy or not. The CHMP has no guideline on these companion diagnostics, and it will not have any in the near future because of the separation of the remits in the two groups. So when a company develops a new drug with a biomarker, I would say, or companion diagnostics, it has to do some things on its own and discuss with the agency in some part. So the discovery is all the sponsor's role, the proof of concept also, although at that stage the company is free to come, it's a voluntary exercise, to the European agency to discuss on the basis of scientific advice what is the best way to develop the product. Then the analytical validation, which was highlighted a couple of minutes ago, is extremely important, is done by the sponsor, could or not, because it's not mandatory right now, get a CE mark from the notified bodies in Europe about the accuracy, reproducibility, robustness, and so on. We don't have any CLIA-like way of standardizing the labs in Europe, but they are, let's say, similar systems. Then the clinical validation is also the duty of the sponsor. Does the test correlate with the clinical outcome in a phase 2, for instance, or even in observational studies? That would be fine. Sensitivity, specificity, predictive values, and so on. And they would only come at the end for the clinical utility. Usually people call it utility rather than validity. That means that does the predictive or prognostic, is the marker predictive or prognostic? That depends on the design of the trials to demonstrate it. And does it correlate with a change in outcome related to the treatment? This is very different from a correlation of a biomarker with the progression of a disease, which we've talked a lot today. If you want to use this kind of biomarker as a tool for drug development, you have to show the correlation with the change in outcome. And sometimes it's completely different. And then finally, the marketing authorization application is going to be discussed at the EMA. So there are really strongly different clinical trial designs depending on whether you want to use and demonstrate the biomarker as a prognostic biomarker, like for instance in this case. I will go relatively quickly, but these slides will be certainly available. You can also, if you are sure that the biomarker negative patients would not benefit from the treatment, would probably not be selected, then you can concentrate on showing a difference between the typical treatment and the biomarker-based treatment in the group of patients with a positive biomarker. That would be the best way to demonstrate the prognostic value of a biomarker. If you want to go for clinical validity, and at some stage you have to do it, the best way to do it is retrospective test. That was also mentioned. So you treat a group of patients with the drug. Maybe that can happen during a trial of the drug versus placebo. That's quite fine, and that's usually how it goes. And then you distinguish responders and non-responders, and you look for the distinction between biomarkers, positive and negative, within the responders and within the non-responders, and you can compare them. And this is usually sufficient to demonstrate clinical validity. Then you need to use it. You have a choice of using either as an enrichment design, so if the demonstration that there is no efficacy of the drug in the biomarker negative patients, if that demonstration is sufficient, usually based on biological plausibility, then you're in a very good situation because you can immediately go for an enriched trial. And both the FDA and the EMA will very much like this kind of design. You don't have to treat the biomarker negative patient, you can simply follow them or not. If not, if the demonstration has not been sufficient so far, then you need the full design, which is very costly in terms of patient and costs also, because you have to show effect of drug versus placebo in both the biomarker positive and the biomarker negative patients, and to show that there is a strong difference. For instance, no difference between treatment and placebo in the biomarker negative patients, and a fine effect in the other group. That usually tends to be avoided because it's really the most costly type of development. So you're better off if you have been able to show the clinical validation first and then go for the enrichment trial. That's what companies are trying to do nowadays, I would say. So there are quite a number of biomarker restricted treatments in Europe and at the FDA. This is a simple list. Most of them are based on oncology, but there are some interesting examples. I think the last interesting examples in terms of a selection of patients is this new product for cystic fibrosis, which you probably have heard about, ivacaftor, was demonstrated in vitro to be active only on a type of mutation that brings the cystic fibrosis protein to the membrane and prevents its activity, and that's where the product was effective. And so the trial was done only in those patients, which are a little group of the cystic fibrosis patients, only 7%, but shown to be very effective. It was not studied in the other cystic fibrosis patients. That was accepted by both FDA, EMA. So they could do a very small study, 40 patients in each group, and then a second study. But the main study was 40 patients in each group, and approval for this product, which has dramatic effect actually on this. They were not given the indication for the other gating mutations because they did not include any patient with another type of gating mutation. I don't know why, but they didn't actually. We have some examples of acceptance of retrospective data in Europe, not at the FDA, but in Europe. This is the most famous case because this kind of curve, which you can discover, I think we had a similar curve shown today, but on the wrong direction because the placebo was going in the right direction at the end. Here it's about a tyrosine kinase inhibitor, so this erlotinib, Iressa, for which the main study that was presented to you was negative, as you can see here, completely negative, but But if you separate the patients according to the real biomarker, which the company didn't want to press at the time, but they knew it was there, they used it as an exploratory marker, although there were a lot of scientific knowledge. That's what you get. From these two curves here, this is what you get. A very significant effect of the treatment with IRESA alone, even better than any kind of chemotherapy versus the group of patients without the mutation. So you need a mutation in the receptor to be able to demonstrate the efficacy of the product. This was accepted by the agency in Europe, and the indication was given based on post-hoc analysis. The product was approved based on post-hoc analysis. This was really a very striking move, but the agency justified itself, I would say, by saying that it was very high statistical evidence, and you can show it, you saw it immediately with your own eyes, that the mutation-related results were repeated in very small subset, but they were kind of repeated, I would say. And especially, and this is the most important point, there was a very strong biological plausibility. The product in vitro could only act on the receptor if there was a mutation, could only block the downstream pathway if there was a mutation, and could not without the mutation. That took, I would say, most of the CHMP members to say yes. On the other hand, at the FDA, this could not have been accepted because the test was not validated. It was just a simple test, I would say, not FDA approved, not validated. So, and the evidence was considered weak. So then the FDA rejected the test. The product was on the market actually at the time for the whole population, but in the meantime it has been suspended in the US because of course the efficacy in general population is low, is not acceptable. It's only active in that subgroup of patients. So this is how you can save, I would say, a kind of failed development. This can happen again, even on the input of the regulators. This is, I think, a good example. Boceprevir, you may know, is a recent product for chronic hepatitis C. It's been approved for the whole population, but there is a strong link, apparently a strong link with the biomarker, which is in this case interleukin 28B. Apparently, if there is a polymorphism in this gene, the patients may not actually respond to the treatment. That was the hint was very strong in the trial, but not demonstrated. So what has happened is that the agency said, yes, you can go ahead with the general indication, but we are waiting for results of subsequent studies. So they are forcing the company to analyze the data in another study based on this discovered biomarker, which is right now not really used. But it may be that next year this study will demonstrate that the product is not active in this kind of patients, so the indication will be restricted in that case. So that can happen many times also. In the end, all these are really imperfect data. I think the regulators understand it very well. But I'd like to quote here one of the most important members of the CHMP, who is also now chair of the scientific advice. He just took my job, and also chair of the biostatistics group. I think this quotation that, from a statistician, from one of the most important members, is very important. So he told us that genomic data, he was talking about genomic data, but you extrapolate to all biomarkers that are generated, can rarely be ignored because these are valid scientific data, even if they are methodologically imperfect, retrospective, and so on. So if you ignore the data, that's an action. And you can err, you can make mistakes simply by ignoring data because they have not been collected in the right way, so you don't want to see them. Sometimes we have some conflicts with the FDA because they don't like this kind of situation here. They want to have everything demonstrated. So this was my feeling on the development of a biomarker linked to a specific drug product. Now, there is a second kind of scenario I'd like to briefly mention. It's called the Qualification opinion procedure. It's relatively new, but it's picking up. And now many companies and consortia are coming to the FDA and the EMA because the FDA has the same procedure. But many of them are actually coming to the EMA to have their biomarker qualified, not for a specific drug product development, but in general for a condition, for part of a disease, for instance. So imagine a consortium developing a new tool It could be a new outcome, a new endpoint, like a patient-reported outcome, a new methodology, a model, or a new biomarker. And they are preparing a plan to develop this biomarker, to use it later on for drug development. And they want to discuss this strategy with the regulators. And they can come to the EMA for what is called the qualification opinion. They need to bring a lot of data. So this is a really extensive scientific procedure where what will be discussed is the value of the biomarker in terms of diagnostic or prognostic or companion diagnostic, so the context of use, as it's called, and then all the elements of the protocol. So whether it's an exploratory or confirmatory design, comparing to the standard of truth, if there is a standard of truth. For the diagnosis, I understand for ALS there is no real standard of truth, so it would be difficult to use that. But this is a point that needs to be discussed. Is there a diagnostic standard of truth to which this biomarker can be compared or not? If not, it's also possible. You have to discuss on the surrogate standard. They don't necessarily have to be used in the current clinical practice, provided they are scientifically valid. So that will be extensively discussed. The endpoints, design, statistical plan also. So you need to be prepared for all this. The final claim will be decided following the procedure. So it may differ from what was proposed by the company, by the consortium, I would say, initially. Who can apply? Anyone, any person. Usually what we get are consortia, networks, public-private partnerships, learned societies, some companies, a few. The goal is to speed up and optimize the drug development based on a qualification of a priori qualification of a biomarker. Actually this procedure ends up in two different ways. If there is no agreement because the data are too premature, then it remains confidential, so nothing is made public. On the other hand, if there is agreement on the qualification of the biomarker, then the result, the opinion is made public. And for everyone to see on the website, for instance. I have a few examples. So the numbers first. This procedure is actually becoming very popular, I would say, among consortia. So for the last 2 or 3 years, about 10 to 12 different procedures have come up. There was a lag period, I would say. And the FDA is following approximately the same track, I would say, with their procedure. Few examples of what has been qualified so far. In 2009, the first procedure, and I was strongly involved in that one because I used to be a nephrologist, was to qualify the biomarker for preclinical research. So now we have a number of these urinary biomarker of nephrotoxicity, like KIM1, total protein, clusterin, cystatin C in the urine, that can be used in preclinical development. And if a company uses them because they have been agreed by the regulators, they can detect kidney toxicology, kidney damage at early stages. There was a follow-up by an academic group, actually, Healthy. The first one, yes, I want to mention, was proposed by CPATH, so the Critical Path Initiative in the States. The PSTC consortium came and developed this, and this was the subject of a couple of papers in Nature Biotech. In 2011, 3 interesting qualifications in Alzheimer's disease. What was qualified and what is now accepted for any future drug development are 3 different types of markers for early stage Alzheimer's disease. So either the CSF signature or PET imaging or even this measure of hippocampal atrophy by MRI can now be considered valid tools to detect progression of patients from the early stages, even from the minimal cognitive impairment, to the Alzheimer's stage. So companies are now able to develop product at very early stage of Alzheimer's disease, which previously were not able to because this entity was not really accepted by the regulators. So now, and this is also accepted at the FDA. So now these enrichment trials can start, and some of them have started actually, in this early stage Alzheimer's disease. And this year, there's a very interesting one I'd like to make. The first one from Novartis is a model of statistical methodology, so probably too complicated for the common people in the room here, but interesting. The second one was also proposed by CPATH. It's called a disease progression and trial evaluation model that fits to mild to moderate Alzheimer's disease. And what they've done with CPATH is collect a huge number of data from clinical trials, from observational studies, active arms, placebo arms, everything together, trying to correlate everything into the progression of the disease from the early stages, not the late stage, and not the minimal cognitive impairment, so in middle. So based on they could identify 4 covariates that actually matter in the development, in the progression of the disease, so baseline MMSE, ApoE4 status, gender, and age. And if you put all this together, you have a model that actually predicts extremely well the progression of all patients, both in the treated arms and untreated arms. So now you can refer to this to power, for instance, clinical trials, to take go, no-go decisions at interim analysis, to assess a disease-modifying versus a symptomatic treatment, and this is important for Alzheimer's, to run post-hoc hypothesis, not to replace the placebo arm so far, but maybe in 2 years from now, the next request will be, can we use this model to replace a placebo arm? And why not? So this could very well happen. This is, for instance, quoted from, and you can read this is public knowledge. You can go to the EMA website and you read this qualification procedure. I think this one is extremely interesting. This is, for instance, a model of a study that has not been run. It's a model, virtual study, with immediate treatment of mild Alzheimer's disease versus delayed treatment. And you can compare the effect and see if your product is disease modifying or not. If the curves run in parallel when you start the treatment with a delayed onset, then it's not disease modifying, it's just symptomatic. Okay, so yes, what I wanted to show you are these 2 different approaches for biomarkers with the regulators, both of them strongly scientifically based, but very different. So the first one is the typical biomarker for one specific drug development, which you may call a companion diagnostic or a surrogate endpoints. I've not discussed the surrogate endpoints, but that would be similar. But I don't think in this kind of disease that we are talking today, we're not really concerned with surrogate endpoints. The patient progresses too quickly. We have a lot of endpoints. So the issue is in the biomarkers. So they need to be extensively validated before marketing authorization, and I've tried to show you the different steps of this validation and different scenarios of the clinical trials and the context of use. Secondly, if there is a broader interest, for instance, from a consortia, a group of academics, they can think about this qualification opinion, which is really an open platform for discussion of biomarkers, usually involves multiple stakeholders, and also come and try to have something qualified at early stages before drug development actually starts in this particular area. These are the 2 points I wanted to highlight today. Thank you.